Hi, I'm Sohail and today I'm going to talk about Calvin cycle. In this lecture, I will focus on carbon dioxide uptake, carbon reduction and ribulose biphosphate regeneration. Plenty of energy absorbed from photons is captured in the NADPH and ATP molecules produced in the light dependent reactions of photosynthesis. The energy and electrons and protons provided by NADPH are required to produce sugars from carbon dioxide molecules in light independent reactions. The reactions using NADPH and ATP to fix carbon dioxide occur in a circuit known as Calvin cycle. Calvin cycle is also known as carbon fixation, light independent phase, dark reaction and C3 pathway. This is the net equation of Calvin cycle. It has 6 full turns of the cycle, 6 molecules of carbon dioxide enter, 1 glucose molecule is produced and 6 molecules of ribulose biphosphate are regenerated. The 6 turns of the cycle produces 2 glyceraldehyde 3-phosphate that can be used to make 1 glucose molecule. So, the 6 turns of the cycle incorporated 6 carbon dioxide molecules providing enough carbon to synthesize 1 glucose molecule. In carbon fixation, the energy of ATP and NADPH is used in the formation of organic molecules from carbon dioxide. Carbon fixation that occurs in stroma of chloroplast in a sequence of 13 reactions that is known as Calvin cycle. The 13 reactions of Calvin cycle are divided into 3 phases, that is carbon dioxide uptake or carbon fixation, carbon reduction and ribulose biphosphate regeneration. So, this is carbon fixation phase 1, phase 2 is carbon reduction and phase 3 is regeneration of ribulose biphosphate. All 13 enzymes that catalyze the steps in Calvin cycle, they are located in stroma of chloroplast. The first phase of Calvin cycle, it consists of a single reaction in which a molecule of carbon dioxide reacts with ribulose biphosphate, that is 5 carbon compound. This reaction is catalyzed by the enzyme that is called ribulose biphosphate carboxylase or oxygenase, that is also known as RuBisco. The chloroplast contains more RuBisco enzymes than any other protein, and the RuBisco may be most abundant proteins in the biosphere. The product of this reaction is unstable intermediate compound which immediately breaks down into 2 molecules of phosphoglycerate. Each phosphoglycerate contains 3 carbon atoms. The carbon that was originally part of carbon dioxide is now the part of carbon skeleton. So, the carbon has been fixed. The Calvin cycle is also known as C3 pathway because the product of initial carbon fixation is a 3 carbon compound, and plants that initially fix carbon in this way, they are called C3 plants. The second phase of Calvin cycle that consists of 2 steps in which energy and reducing power from ATP and NADPH both are produced during light dependent reactions. They are used to convert the phosphoglycerate molecules to glyceraldehyde 3-phosphate. For every 6 carbons that enter the cycle as carbon dioxide, 6 carbon can leave the system as 2 molecules of glyceraldehyde 3-phosphate to be used in carbohydrate synthesis. Each of these 3 carbon molecules of glyceraldehyde 3-phosphate is essentially half a hexose 6 carbon sugar molecule. The reactions of 2 molecules of glyceraldehyde 3-phosphate is exergonic and leads to the formation of glucose or fructose. In some plants, glucose and fructose are then joined to produce sucrose. The plant cell also uses glucose to produce starch or cellulose. So, 2 glyceraldehyde 3-phosphate molecules are removed from the cycle and 10 glyceraldehyde 3-phosphate molecules they remain, and they represents a total of 30 carbon atoms. So, each glyceraldehyde 3-phosphate molecule has 3 carbon atoms and 10 glyceraldehyde 3-phosphate molecules, they represent 30 carbon atoms. Through a series of 10 reactions that make up the third phase of Calvin cycle, these 30 carbons and their associated atoms become rearranged into 6 molecules of ribulose biphosphate, and each ribulose biphosphate is a 5 carbon molecule. So, each ribulose biphosphate is phosphorylated by ATP into ribulose 1,5 biphosphate. These ribulose 1,5 biphosphate molecules begin the process of carbon fixation and eventual glyceraldehyde 3-phosphate production once again. Let us watch this slideshow.